Okay, now the going gets tough. So I want you all to listen to what I have to say, because I'm giving you something new. That PCR assay and that DNA blot assay, Derek and Anna, no one does that anymore. What do you think the reason for that is, Mike?
We have better technology.
Why do you think? Where's your microphone? Yeah, we always have, but why do you think? Who's doing these tests? Who does these tests? Where are they usually done?
Genetic counselors.
Where are they done?
Laboratories.
Where are they done? Where's the laboratory at?
Research facilities.
I don't think so. Where's the laboratory at, do you think, that does these tests? What do you think, Eden? Police station for testing disease genes? A police station? Where do you think these tests are carried out?
In
Julie?
hospitals?
In hospitals. Now, if they're going to be done in hospitals, do you think that they have to be cheap? Yeah. You think they have to be simple? They have to be robust? Well, that's the point. So the technology does evolve, but they have to be done in a much simpler way. Do you think they want to use radioactivity in the hospital? No, they don't want to use anything which is radioactive. They don't have to. They need a simple test that technicians can carry out. So the technology evolved, and I'm going to show how it evolved further, by using these things called allele-specific oligonucleotide probes. That's a big word, isn't it, Anna? Allele-specific oligonucleotide probes. Derek, what's an oligonucleotide? You're a biochemistry major. What's a nucleotide? Speak up.
Monomer of DNA.
Monomer of DNA. What's an oligonucleotide? Speak up. What's an oligonucleotide? Did you ever hear that word? What's an oligonucleotide? Think it's a big, long DNA molecule? A polymer? What's a polymer of DNA?
A short sequence.
A short sequence is an oligonucleotide. It's a short sequence. There they are. Oligonucleotides are short sequences. The DNA molecule, Chris, is a polymer. Eric, it's a polymer. It's a very long sequence of nucleotides. So we have nucleotides. We have oligonucleotides of a short sequence, 10, 20, 30, 40 nucleotides. And then we have polymers of millions of nucleotides all strung together by phosphodiester bonds. These oligonucleotides are very powerful. The reason why they're powerful, Max, tell me something about the double helix. Elaborate on the double helix for me. Take the microphone. What's one feature of the double helix?
The two strands are anti-parallel.
What's another feature?
The bases are complementary.
What's another feature? What holds the bases together?
Hydrogen bonds.
Do you think that a polymer of 1 billion nucleotides in a row and 1 billion hydrogen bonds would be more stable than something that's an oligonucleotide of 30 and only has 30 hydrogen bonds?
Yeah.
If I heated up the long thing and the short thing, what would need the higher temperature?
The long one.
Yeah, absolutely. So if you have a DNA molecule that's huge, in order to break those hydrogen bonds, you need, let's just say, a lot of energy. High temperature. But if you have something that's really short, there's not as many bonds holding it together, you need a lower temperature. What that means is that if you take the DNA apart, it can reform again. Do you agree with that, Stephen? Because it can reform again. But there's one really neat feature of these little tiny things. At most temperatures, Kathy, and other Kathy's gone, at most temperatures, Kathy, that we work at, a little short thing is not so stable because there's not as many bonds holding it together. Does that make sense to you? Okay. So in reality, what that means is that if you have a big, long DNA molecule, millions of nucleotides, and you take DNA from one person and a DNA from another person, and you try and bring them back together again, they'll come back together again even though they may have a nucleotide or a 3 or 4 or 5 or 6 that are different from each other. They can still reform. But these little short things, because they have so few hydrogens to hold them together, if there's any one difference between the two, you can distinguish them on the basis of temperature. And I know that's a mouthful, but let me show you what I mean. And this is very powerful. Let's say I have two alleles. There's allele 1 and there's its complement. This is a double-stranded DNA molecule. This long one is in the genome. This short one is, let's just say, a probe that we synthesized. Notice that in order for this to form a double-stranded molecule, it has to be absolutely 100% complementary under certain conditions. Even if I heat it a little bit, it'll still stay in a double-stranded state. On the other hand, here's allele 2. Notice that allele 2 has a single nucleotide polymorphism at some sequence, which means when I bring these together, here's the DNA in the genome of allele 1, here's allele 2 as a probe, there's a mismatch, meaning you can't get a complementary base pair because it's different. In other words, the C isn't going to form with an A. Because this is short, if I raise the temperature, the whole thing falls apart. Completely falls apart. Because there's so few hydrogen bonds holding this thing together. The point here is, conceptually, is you can use this thing called allele-specific oligonucleotides, ASO, to distinguish between one nucleotide difference in alleles or even individuals. I'm going to say that again. You can use these very short oligonucleotides to distinguish between two different alleles because of the properties of the double helical molecule DNA. Meaning, if you only have a few hydrogen bonds, even if there's one base pair which doesn't, called mismatch, doesn't form a complementary pair, under conditions in a laboratory, you can cause them never to form a double helix. Is that clear, Stephen? So you adjust the conditions in the lab, and you say the only thing that can form with these things is a perfect hybrid, a perfect match, a perfect duplex, a perfect double helix. If there's even one nucleotide difference between them, they won't form. Is that clear, Chris? Eric, is that clear? This is in a laboratory now. We're not doing this in the cell. So you can adjust these conditions. So that means if I have allele 1 and allele 2, and I know what the sequences are, because you have to sequence them, and I know what the difference is, and I synthesize a probe, an oligonucleotide probe for both of them, it means that I can use allele 1 probe to detect only allele 1 in a genome. And under the conditions in which I get these things to reform a helix, allele 2 will not form with allele 1. And so that means very nicely I have an assay that's very simple. And if I put something on these probes, not a radioactive thing, but if, let's say I put something that can give a color, and I'm not going to tell you what that is, but if you put something on there that can give a color, notice what I can do. And so here's how they're used. I have oligonucleotide for one allele, ASO for a different allele. I have a number of individuals in a population. I take their DNA and I isolate it from individual 1, individual 2, individual 3. I just isolate the DNA from their cheek or their blood or from something. Because I know the sequence of the gene that the ASO comes from. Is that clear, Tiger? These probes are specific for a specific gene. The gene that has the difference. The difference is the marker. We start with a SNP in a particular gene. Okay? Since I know the sequence of that, I can design a polymerase chain reaction to amplify that particular region. Is that clear, Katie? Because I choose primers in that gene outside that polymorphism, so I can amplify it. So I amplify it in individual 1, individual 2, individual 3. We're just amplifying the region that we're trying to test for, let's say some disease gene. And I'll give you examples of this in a second. It's very powerful. And I put the DNA that I've amplified on a little filter. There's filter, and you know about putting DNA on filters because we do that to screen libraries and other kinds of things. I put the DNA from person 1, person 2, person 3 on 2 filters. It's kind of like a replica because I'm going to test them with the allele 1. I'm going to test them with the allele 2. And of course, the DNA, Julie, is denatured because you can't form a hybrid between 2 double-stranded DNA molecules. Michelle, am I clear? Tell me if you're not, because this is so powerful and so neat. Wait till I show you some results in a second. So now I have on these filter papers individual 1, individual 2, individual 3. I take those filter papers and I hybridize each with both probes. Because I want to assay for allele 1, I want to assay for allele 2. And the results then, when they hybridize with some means of detection and it's not radioactive, what you can see here is very simply that allele 1 hybridized to this person, that person's DNA, and not that one. Allele 2 didn't hybridize to that one, but hybridized to this person and that one. And now you know immediately the genotype of these individuals. This individual has to be homozygous for allele number 1. This individual has to be heterozygous for alleles 2 and 1. And this individual has to be homozygous for allele 2. And that's a very simple assay for a disease gene in a hospital, done non-radioactively, done really quickly using the polymerase chain reaction. Remember, you isolate the DNA from these individuals. You know what gene you're testing, like sickle cell anemia. You amplify up the region that has the difference. You know where the mutation is. You know where the SNP is. You amplify it up from each individual using polymerase chain reaction. You take the DNA from those individuals and you put it on these little filter papers. You replicate them. You denature them. And you anneal or hybridize to each of these very short oligonucleotide probes under conditions in which a perfect double helix has to form. If there's one mismatch, you get nothing. And so now you can distinguish between whether the individual has allele 1, allele 2, or both. And that's exactly the basis of the technique. Julie.
So does that mean you can only run this test with alleles that are already known?
Yeah, you can only run this test for alleles that are already known. Very good question.
Would you need probes then with radioactivity to detect new ones?
You'd have to find the new ones. And you'd find the new ones by saying, here's sickle cell anemia well, that's a bad example. Here's cystic fibrosis in a new family. You can isolate the diseased cystic fibrosis gene because you know what the normal gene is. You sequence that gene and you say, it's a polymorphism in a different region. Or you scan the entire gene at one time. Meaning you could do this, and I'll show you how that's done in a second. You can just, for example, say if it's a small gene, maybe there's a mutation at every single solitary coding sequence in the gene, and you just make 1,000 probes for that gene that covers any part of the gene. So there's a lot of ways in which you can do this. Okay? Remember what I said well, you probably don't remember, but I did. I said this on last Thursday. Many of these disease genes are the result of founder effects. Remember I said that? That they happen way back when and they're passed on. So many of these disease genes, Nancy, are passed on through millions of years. So you only need one test. Like cystic fibrosis, really 70% you need one test. Sickle cell anemia, there's only one cause of sickle cell anemia. I mean, you only need one test. It's a very specific change in a DNA. You can have a lot of different forms of hemophilia, but you only have one sickle cell anemia form of hemophilia. And that's due to a very specific base pair change and a very specific amino acid sequence change. Let me show you some results. Here's a very neat result that we'll talk about in a couple of weeks. Here's in vitro fertilization, which has been in the news, unfortunately, with this octuplet case over the last week or so, which we'll talk about in a couple of weeks. Here's in vitro. This is a family of individuals that is known to be carrying cystic fibrosis. The parents underwent in vitro fertilization. A part of the then 8-celled embryo, one cell is taken by pre-implantation genetic screening, PGS. The DNA is taken out of each of the embryos, and there's many of them. The cystic fibrosis region is amplified up with PCR. We have allele-specific oligonucleotide probes for the normal and the wild type cystic fibrosis gene. And now you look on this little filter paper. This embryo is homozygous for cystic fibrosis. This embryo is normal. This embryo is cystic fibrosis. Here's heterozygote. Here's a wild type. Here's cystic fibrosis. And now you pick this embryo, maybe that embryo, and implantate in the mom. And generally, well, not generally, the kids will not have cystic fibrosis. And so that's the way this is used in pre-implantation genetic screening. Another example is sickle cell anemia. It's exactly the same procedure using these allele-specific oligonucleotide probes. Here's an individual that is heterozygous. Here's an individual child which is normal. Here's heterozygotes. You can use that to follow pedigrees. Pre-implantation, you can do anything you want with it. It's extremely powerful. Should DNA testing be carried out on every individual born in the United States? Yes or no? This is really neat stuff. Okay. Yes. Who said yes? Julie.
Because then you can choose whether your kid has a disease or not. And I mean, if the disease is going to be
Candy bar. Go ahead. I'm running out of sugar here. Go ahead. No. Go
if the
ahead.
disease is going to be devastating, you obviously wouldn't want to live with it, and you wouldn't want to make your kids live with it.
But you think that everyone should be screened? It should be mandatory? Everybody should be screened?
Well, yeah, then they'd have a choice. If they don't want to choose to, then okay, but
Okay.
at least they'd know.
Well, we've talked about this, and we're going to talk about it again in a few weeks. Who said no? Dora.
I thought it should be a choice if they wanted to have their kid DNA tested or not, because once you find out, you might be like under a lot of pressure or like stress because you know that they carry a gene that.
Remember, I asked you this question a few weeks ago, but you might not remember. Have you ever been tested for any genetic defect?
Not
Have you?
that I know of.
Have you, Tiger? Yeah. You're positive.
I believe that you said that in an earlier
You've
class.
all been tested. It's mandatory in the state of California, 160 genetic diseases. Parents have no choice. They only can opt out on religious grounds. But generally, they're only genetic diseases for which they can be, I won't say treated because it's a different, but let's just use that they can be treated. Like phenylketonuria. Change the diet, the baby's born and completely healthy. So it's not for things that you can't effect a treatment and help. It's only for things in which you know that if you immediately change the diet or do something, you can have a healthy baby that lives normally forever. A rose is a rose is a rose is a rose. Okay.
But
go ahead. No question? Okay. DNA testing results should be made widely available, meaning to everybody. Yeah. Stop. Well, that's pretty. Is anyone going to volunteer who said yes, or is it just pulling my leg? Elaine, you pulling my leg? You would never do that, would you? Never. Is anyone who wants to take, Kathy, you want to take the opposite point of view? Good for you.
Did you mean to that, that individual's results can only be seen by that individual?
No, I mean it can be seen by anybody. Change your mind. Okay, good. Yeah, I think that should be a private conversation between you and whomever. Okay. Is it clear what I've said so far? Let me add a footnote here. When I talk about these things called DNA chips that scan all the genes in your genome at one time, you with me, Maura? The basis of doing that technically is ASO. When you think about these DNA chips, think about these things called ASOs, because that's just the conceptual basis. The difference is when you do the ASO like I showed you, you're doing one gene at one time. When you do these DNA chips, you're doing thousands of genes at one time with, thousands of probes, essentially. But that's the way in which it works. And we have one individual in this room that spent a considerable amount of her time helping to develop these kinds of DNA chips for doing that. And she's right back there. And so if you want to know about how this is really done in real world and how people spend millions and millions of dollars developing these tests, ask Aicha, because she worked very hard in helping to help a company called Affymetrix, which makes these chips, develop these things. And when I talk about the chips, here is one. This is what they look like. So when we talk about these things called DNA chips, or you read about them in your book, that's a DNA chip. That's a chip that has 22,000 genes on it that can be used to test for 22,000 different things at one time. It's very simple. Okay, now let me digress for a second. So you can have mutations in genes due to single nucleotide polymorphisms. I'm still talking about genes. But because of the peculiar nature of the genetic code, and because of the peculiar nature, Hiromi, of proteins that are made by specific genes, sometimes a gene has a codon that goes like CAG, 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 because the amino acid X is repeated XXXX in a row in the protein. It's not a very frequent occurrence, but it can happen. So in a sense, and I don't want you to misunderstand what I'm saying now, so listen carefully, Max. In a sense, it's like a VNTR, but it's not a VNTR. It's a repeat of a codon within a gene. So it's kind of like a simple sequence repeat, but it's within a gene. Do you understand that? And it's unique to that gene because it's a particular codon for that protein that's specified by that gene. But the VNTRs are like an accordion, right? They can expand in individuals and they can contract, correct? So if you have this situation in a gene, can you imagine, Brian, that it could cause some devastating results? Yeah, because if there's mistakes that can allow these VNTRs to expand, these same kinds of mistakes can allow these codons that are repeated in a gene to expand as well. And what do you think the consequence of that would be, Brian? I can't hear you.
Make a protein that doesn't work?
Yeah, and what would it be as a manifestation? How would that be manifested in humans?
You wouldn't have the right protein.
And what would it cause?
Some kind of disease.
Yeah, it would cause some kind of mutation or disease. And this is devastating. And when this happens, in many cases, the gene codes for a dominant disease because the protein is so screwed up, and I'm not going to get into the reasons why, it acts in a dominant way. And what you can see here is that there's many of these particular disease genes, and here they are, that have this repeated codon. This is a codon in the gene. And one of them is this thing called Huntington's disease. And I think all of you have heard of Huntington's chorea. It's a devastating disease. It's dominant. 100% death at 45, or 50, or 60, or 30. There's no escaping. And I'm telling you, if you've ever read any books on Huntington's disease, it's devastating because you lose all neural activity and it slowly degenerates. And if you have a family in which Huntington's disease has been transmitted, it's pretty scary to know whether or not you have it or not. But of course, now there's tests. So if, God forbid, you came from a family that had Huntington's disease, how many of you by hands would want to be tested and want to know, or just would want to just luck of the draw? Can I see the hands up? Thank you. How many would not? Oh, it's pretty good. I think I'd want to know too, so you could prepare yourself for this and live a full life prior to when this would happen. So the way in which the disease works is you start expanding. And the more expansion that you have, the more devastating the disease. And furthermore, can you imagine in pedigrees, though, Max, that you have this disease allele and you think, oh my God, I'm going to pass it on to the next generation, but it's possible that as it expanded, it could contract too. And so the inheritance of these disease genes are really murky. They don't follow a normal Mendelian pattern because it can be normal, 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 mild, devastating, normal again, even though you carry the thing. Mild, mild, devastating, normal, normal, because these repeats are expanding and contracting in each generation depending upon the DNA replication process that's going on. Most of the cases, this doesn't happen, obviously, or we would all be dead, but they cause all kinds of difficult diseases. So what I've talked about in summary here is that we have single nucleotide polymorphisms inside Mendelian genes. I've said that only about 3,000 Mendelian genes are known in the human genome, and those single nucleotide polymorphisms can be used to trace the gene from using these tests. And here we have an example, though, of a little variable number repeat of a codon. And in that situation, they can expand and cause a defect, a disease. And again, you could use those as markers, obviously, because you know where they are. And you can use a variety of different ways in which to assay for them. And you know those ways. And here's this weird inheritance of this. So how do you then detect these single nucleotide polymorphisms in individuals? And how do you go about doing this? Well, you know this, and I'm going to go rapidly over this because there's no point getting into it in depth. The only thing that you need to know, Thomas, is that in order to do any of this, you need to have the clone first, as I've said, and the sequence. Can't do any of this without a knowledge of the clone and the sequence. And so here we have a single nucleotide polymorphism. This can be used now not simply for a disease gene, but you can use it to identify individuals. And in this situation, the single nucleotide polymorphism eliminated a restriction site here. And so this individual in this region of the genome with digestion or with the restriction enzyme would get 2 bands. This individual would get 1, 2, 3 bands. And so you can distinguish the 2 of them between their DNA fingerprinting pattern. And the only way you can find that is either by using the polymerase chain reaction or a DNA blot, in a sense. Those are the polymorphisms. In this case, we're not using ASOs. In this case, we're using restriction length fragment polymorphisms to distinguish between those 2 individuals. And as I said, for the mitochondrial genome, you can have the same thing, and you can follow them. You can have single nucleotide polymorphisms in the mitochondrial genome, and those polymorphisms will be followed a maternal inheritance. Furthermore, as I've said, you can have a single nucleotide polymorphism, and you can detect it by PCR. And in this case, you would use primers outside the region where the mutation occurred. You'd amplify that region up, and then you could digest the amplified products. And here again, you can see homozygous, homozygous, heterozygous, 2 different alleles. And in this situation, you might also be able to use allele-specific oligonucleotide probes. Here's a VNTR, same situation, using PCR. This is what you're going to discuss again tomorrow in your discussion. But again, conserved regions that are every allele has the conserved region. No difference between alleles. The difference in this case is these simple sequence repeats. It's between them, and each one will produce a different size on a gel because it's an accordion. And furthermore, you can then follow those by a pedigree, and you can follow the inheritance of your parents, siblings, great-grandparents, or whatever, simply on the basis of these sizes. Stephen, is that clear? Is it clear, Kate? Okay, it's clear to everybody. I mean, we've gone through this a million times. And you can use these VNTRs to identify individuals. This is one locus. Why? Because there's only 2 bands, just like your DNA fingerprint in class. And so here are the different individuals. This one's homozygous, heterozygous, 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 heterozygous. You can go through the whole thing. And you can use this to identify, again, individuals. And here is the Tsar family that was brutally killed by the Bolsheviks when they came into power. And you can use these these simple tandem repeats to identify that they were indeed the Tsar, the Tsarina, and their children. And that was hot in the news about 8 or 9 years ago. And again, mother's chromosome, father's chromosome, markers for each. And here are these different tandem repeats, 15, 16, 15, 16, 8, 10. Those are the numbers of each repeat in those individuals, and you can use those. This same thing was used, in essence, for showing that Sally Hemings was the mother of many of Thomas Jefferson's children up until this day. The same kind of DNA testing to show this relationship. Here's a 2 VNTR situation. So in this case, we're using 2 different loci, VNTR-A, VNTR-B. How do you know that? Because you have 4 bands. Remember, 2 alleles per individual. 2 individuals, you can have 4 different forms of the same allele. The more loci you use, the more accurate pinpointing to individuality. We're not talking about disease genes now, we're talking about individuality. And in criminal forensics, they use typically 15. 15 different simple sequence repeats. And so here's a 3 locus. Each of these have a different sequence. You'd use 3 pairs of primers collectively. You would amplify these 3 loci up collectively. And you can see that the gel is a little bit more complicated, but you can get, in a sense, 2 different forms in this person, 2 different forms in that person, 2 different forms in that person. So you can have 6 different bands in each of these individuals because you're testing for 3. And again, with DNA criminality, for forensic testing, you test multiple probes. And you'll talk about that tomorrow. And so clearly, here's 1, 2, 3, 4, 5, 6, 7, 8, 9 different VNTRs, all done separately. You can either do them together or separately. You can see from this gel that doing them separately is easier to interpret. That's generally the way it's done, separately, because you can interpret the results more. But you can see that clearly, I don't think there's any question of which suspect is the one probably responsible for the crime because of these matches in almost every single solitary case. And then, as you'll see tomorrow, you can calculate a probability on the basis of this. So, you with me, Chris? This is very powerful stuff. Tragically, almost 3,000 people died on 9/11. Hopefully that will never happen again in my lifetime or in yours. But the point here is that 3,000 people were blown to smithereens by a bunch of misguided terrorists for whatever reasons that they felt were important to do this, horrific reasons. But we have 3,000 people who were blown to smithereens, whose families were completely devastated, completely. Can't even imagine how their lives were completely wrecked forever, never to be really normal again. But they wanted something tangible, and that is something to identify that their loved one was there. And so these 300 pounds of remains were tested using DNA tests over the last 8 years. And you can see that many thousand, well, 1,000 or so of them were identified using DNA testing. Can you imagine how difficult that is? Using like little 1 speck of a tissue or 1 little part of a bone or 1 little fragment. And the way they were identified was to get from the families of these victims of this horrific crime their comb, their toothbrush, their hairbrush, and DNA was taken off of that from their skin cells, and a bank was put together, and then they were matched with all these random DNA tests that were done on all these different fragments, of which they put 1 little fragment that they found in 1 little bag. All these things are separated. And furthermore, these families of these victims did not want, under any circumstances, the remains of their loved ones to be mixed in with the terrorists. In fact, many of them, and I don't blame them, wanted the remains of the terrorists so they could do whatever because they were so angry and remain angry. And so the FBI and the NYPD and a variety of other testing services actually went through these 300 pounds and were able to separate bit by bit, tissue by tissue, all of the terrorists that they could identify from the remains of all of the victims. Isn't that amazing? And put it in a separate place. And they did all that by DNA testing. And how did they know that the terrorists were the terrorists? Well, they couldn't get any of the family members' DNA because, of course, no one ever admitted that these people did anything. But they had the rental cars, they had the hotel rooms, they had cigarette butts, so they had traces of these individuals, and they were able to identify, as you can see here, 13 of the 19 terrorists. Now, some of these individuals will never be able to be identified because, tragically, not the terrorists, but the victims, tragically the victims were just blown to smithereens. I mean, there's absolutely no trace of them. And so they're still continuing these studies, but again, this is the power of DNA testing in this tragic, horrible, horrific situation. But in this particular case, it gave many of the victims' families some peace of mind or something maybe tangible that they could put the remains to rest and have memorial, or maybe someone would have closure because they really never knew whether their loved one was there or not. They just disappeared, and they are confident now that forever those individuals would not be mixed with the people that perpetuated this horrendous crime. And so that is the power of DNA testing. And here are all the different uses. Aicha and Daisy and Kelly have discussed those, and many of you can review that. Okay, let me switch topics just a bit. The technology is now emerging because of the sequence of the human genome that you can now scan for single nucleotide polymorphisms across the entire genome at one time. Is that clear? So you don't have to just look one locus at a time. You can look in thousands of loci at a time. Is that clear, Derek? You with me? You drifting? Don't drift. Okay? Don't drift. Stay awake. This is the most cutting-edge stuff you're ever going to get at this university. I trust you. This is right in your home. This stuff affects us. So you can scan across the whole genome using these things called DNA chips, or what will now replace them probably within 5 years, that is whole genome sequencing. You know, we'll just sequence very cheaply and very rapidly all of our genomes. And as I said to you last time, it took 15 years and $3 billion to sequence the human genome. I can sequence Thomas's DNA in my laboratory in 4 days for less than $100,000. It's not easy, but it's easy in comparison because there's been all these years of developing all this technology, and it's gotten really cheap. So there's two things that you can do with this. You can assay then all the single nucleotide polymorphisms across populations. I mean, what if we sequenced everybody in this room and we put it in a database? I'd be able to catalog all the single nucleotide polymorphisms of everybody in this room. And we'd find differences, okay? We'd find a lot of similarities, but we'd find differences. So we can do that to identify individuals. But we could also identify the single nucleotide polymorphisms with known disease genes. And I'll show you how that's done in a second. Across the entire genome. We can associate these single nucleotide polymorphisms with disease markers. And furthermore, we can also use this information to say that someone came from a particular region of the world or a particular, as I'll show you, county in a particular country. That's how powerful this is. Someone can trace your ancestry back to a particular neighborhood in a particular city in Europe, down to the neighborhood. That's how powerful this is. And so by using a whole genome scan, as they say, of these single nucleotide polymorphisms, we can do all kinds of different things. So let's just talk about disease genes for a second. If you know what the disease is, and if you know that the disease is associated, I said, with a certain single nucleotide polymorphism, you can use that single nucleotide polymorphism as a marker for that disease. And of course, each disease might have a different single nucleotide polymorphism marker, and across the whole genome you can scan for all kinds of stuff. Not only diseases, but eye color, skin color, all kinds of different stuff. Any trait that you associate with a single nucleotide polymorphism, you can start assaying for. And how do you associate a trait with a single nucleotide polymorphism? You do it in what is known as big population studies, meaning there's a population here, let's say, that is green, a population here that is red, and you look for a correlation between a certain polymorphism in the green population that differs from the red population with some probability. It's never 100%. You know, it could be 5%, could be 15%, could be 30%, could be 50%. The bigger the population you assay, the more confident you are of the association between the marker and the phenotype. And this is a hot area of human genetics. Everyone is doing these big population studies for skin color, eye color, heart disease susceptibility, obesity, anorexia, everything you could ever imagine under the sun. People are doing these big population studies. And those are called association studies. And so one wants to associate, again, Julie, with a specific probability, some low, some high, that this SNP associates with. This is not something like it's a marker in the gene or next to the gene. This is just from a population study association. And so there's this company called 23andMe, founded by the wife of the guy who started Google. This particular company, for $399, will scan your genome for a certain number of SNPs, and those SNPs were shown by these association studies done by other people, taken off of the net, which I'll show you, grabbed by the company, and then when they scan your DNA, they say, oh, you have a 5% chance of having this, you have a 10% chance of having that, you have a 20% chance of having that, et cetera, et cetera. Any of you can do this. On the other hand, there's another company that's a little bit more robust. Costs more money. Costs a couple $100,000. They'll scan your entire genome. In fact, they'll sequence it. So for $200,000 or $300,000, you can send your DNA to this company. They will get the entire sequence of your genome, and they'll scan it with respect to these SNPs that other people have shown are associated with different diseases, traits, or whatever. So it's this personal genomics, the age of the personal genomics. And it's done by these whole genome. Where's the chip? Can you throw it? It's done by these whole genome chips, and it's done using your DNA, hybridized to, in this case, on the chip would be the single nucleotide polymorphism that's associated with all of the traits that you're looking at. Okay? So on this chip might be 3,000 single nucleotide polymorphisms that are associated with 3,000 different traits. Is that clear? And then what they would do is take your DNA, now your whole DNA. They take Kate's DNA and hybridize it to the chip. So in a sense, the allele-specific oligonucleotide is on the chip, and what's being hybridized is your whole DNA. That's the opposite of what I showed you for the ASO. In the ASO, your DNA was on the filter, and the probe was the little oligonucleotide. In this case, the little oligonucleotides are on the chip, but they're thousands at a time. Is it clear, Max? You're positive. This is the most cutting-edge stuff in human genomics. This is changing everything. This is going to change everything in which we do from a medical and also from an ancestral point of view, as you will see. So there's your little chip. In a sense, you can assay for this in the same way. I'm not going to go through it. But in a sense, you have an individual that's, let's say, homozygous at this locus, does not have that SNP, heterozygous here. You can just follow it. It's like an ASO. You can read the slide yourself. But this happens 1,000 times on that chip. And a computer will take those signals over each of these little regions, because each part of this chip, each part of that chip will have 1,000 or 10,000 of these things. And then by using computer graphics and everything, they'll translate it. And I'll show you one of those in a few minutes. And so we get the personal genome. And now we start saying 5% correlation with heart disease, 3% with obesity, 60% with schizophrenia, 45% with bipolar, et cetera, et cetera. What are you going to do with this stuff when you get it, Brian? Got this big readout. What are you going to do with it?
Look at it
Yeah, but what are you going to? What's it useful for?
for determining my risk of getting a certain disease.
What did I teach you the most important thing that I want you to learn in this class?
Critical thinking.
Okay, what are the problems with this? What are the problems with a company like 23andMe that are going to take your $399 and rip you off? Tell me some problems.
It's only giving you the risk. It's not
It's
necessarily
giving
gonna
you the
happen.
risk. Critical thinking, Brian. Where do they get the data for the risk?
From my DNA.
The data
Well,
that says the risk. Where do they get the data that says there's a 3% risk?
It's compared against other people.
Did you look at those data?
No,
Do you know that those tests that were published were good
No.
or bad?
I don't know.
You're just going to take their word for it that they used this study published in the Journal of Afghanistani Human Genetics, that there's a 3% chance of being associated with this trait, and you're going to say, I have a 3% chance of that. You're going to believe that crap.
No.
Excuse my language. That's the point. What's another problem with these tests, Samantha? Did you do the test? Were you there when they did it?
No.
Was it approved by any agency?
Not that I know.
Does it have any license? Is it approved by the FDA?
It's not approved
Was
by
the
the
lab
FDA.
ever inspected?
No.
How do you know that the test is real or not real? Don't you think there might be a false discovery rate of some kind?
Of course.
Oh, but now I'm 30% susceptible to heart disease. I better go to the doctor. I have the breast cancer gene. I better get a mastectomy. There's some problems with these tests. You guys have an advantage now because all of you know the basis of these things. You can evaluate them. If you took this test, you'd be able to evaluate it. No regulation, no FDA certification, taking published studies from the literature of which you don't know how good the studies are or not because you haven't read them all, and some of them, frankly, are lousy. Might be 300 people, might be 20 people, might be 5. You don't know which ones are good or which ones aren't good. But the point here is that even if you, and here's one, for example, happens to be a good one. This is the kind of test that's used. This is an association test. 14,000 cases, 7,000 of 3,000 controls, and there's your SNP. That's where they're taken from. There's your SNP for coronary heart disease. There's your SNP for Crohn's disease. There's your SNP for hypertension. They don't have a lot of good probability here. You see how small that little green thing is compared to here? The higher the bar, the more probability of correlating the disease with the SNP. But you go to 23andMe, they might use something like this, 2% chance. What are you going to do with it? I don't think there's anything wrong with taking these tests, but I'm not sure what you can do with these tests. But here's where they come from. They come from the web. They come from the genome people, the National Center for the Human Genome Research. They come from published studies that are compiled, that people do these association studies between traits and specific SNPs. And then they're used by all these companies. And they're used by ancestry companies that I'll go into in a few minutes. And so the question really is, how valid are they? How good are they? And so there's a caution here, a big caution. How do you use the information? How good is the test? Who did the tests? What's the false positives on the test? What do you do with the information anyways? Live in a closet the rest of your life because you have a 2% chance of something? So the point here is that they're only predictive, they're only correlations. What if someone, I don't know, I just got a letter from UCLA. They said, Dear Professor Goldberg, you better go onto the internet because all of our medical records at UCLA Hospital were hacked into last week. So all of your Social Security numbers and medical records are now in the hands of a hacker. What if someone hacks into 23andMe and gets a hold of all of your DNA sequence information or your testing information, puts it on the internet or whatever? There's a lot of problems here in terms of discrimination, in terms of privacy, in terms of efficacy of the tests. It's not just simply, oh, I'm going to go on the internet and I'm going to send my DNA sample in and see what happens. And that's what I mean by critical thinking. And of course, if you sequence the genome, then you have the same problems. And this is where it's going. Sequencing the genome because, lo and behold, our friend here, by the name of James Watson, was the first individual that ever had his genome sequenced. And that was sequenced by this company, Know Your Genome. And you can send in again your DNA and get, Ben, you have $300,000 in your bank account? Anyone has $300,000 in their bank account in this room? Someone must. In any case, you can have your sequence. They send back, can you imagine a big book, ATTAAAGCCCCTT. Is really, that makes for good reading. You can stay up there at night and read chapter by chapter. Tonight I'm on chromosome 1. I mean, it's ridiculous if you think about it. But that's what you get. So, but the point is, it is powerful, and you can do a lot with it. And so one of the hottest areas now of human genomics is this thing called the 1,000 Genomes Project. And that's a project which is sequencing 1,000 genomes from different groups around the world and to try and understand all the polymorphisms between these groups and make correlations between these groups and diseases that they're susceptible to, their ancestry, and everything else. So here we go. 1, 2, 3, 4, 5, 6. Oh, God, everyone says yes. You're going to read this thing? How long do you think it'll take you to read chapter 1, Chris? Yeah, exactly. Yeah, I could care less, to be truthful with you, but each one of their own. Okay, let's take a look at this thing. I hope I can play it. Whoop. Darn it. I did that on purpose. Just a second. Here we go. Pull this up.
Plus, the secrets in your DNA. A look thousands of years into the past with just a bit of saliva. Might you be related to Genghis Khan or Jesse James? I put myself under the microscope.
From the global resources of ABC News with Terry Moran, Martin Bashir, and Cynthia McFadden. This is Nightline, December 4th, 2007.
Now to the hidden story of our genes. History tells us that Genghis Khan amassed an empire that stretched across all of Asia. He also had many, many children, so many, in fact, that one estimate suggests as many as 32 million people may have descended from him. So might you be one of them? Well, it's now possible to check. A company in Salt Lake City is building a database that may someday connect us all. And they believe it's one way to change the world. It's one of the first questions we ask. Where are you from?
I'm Russian.
Ako'y Filipino.
I'm Bengali.
I'm Irish. I'm Korean.
Soy cubano.
I am, yes.
National identity has been used to divide people for generations.
African American.
Filipino.
100% Armenian.
But these nationalities may be more closely connected than we ever imagined, as genetic science opens the door to a new understanding of human ancestry. The prospect of learning more about your roots through science is particularly appealing to people like me, who know very little about our ancestry. I do know that my parents were born in what was then India and emigrated to the United Kingdom in 1951. But since I was born in 1963, I've never met any grandparents or any other relatives beyond my brothers and sister. So is it possible that the new science of genetics can help fill the gaps in my own genealogical past?
There's only certain people who have access, and then that's recorded every time they come in and go out.
Dr. Scott Woodward is Chief Scientific Officer of Sorensen Genomics in Salt Lake City. The company, which originally offered paternity testing, is now giving people the chance to trace their lineage by using their own DNA.
We take a look in here, we'll see a portion of the 100,000 samples that we have that we're using to build this database.
For 7 years, they've been building a mitochondrial DNA database. This is DNA that's maternally inherited and the purest form of genetic inheritance because it barely changes through the generations. How sterile is the process?
We keep that very tight. DNA and the amplification process that we go through is very, very sensitive.
I'm assuming I can't touch anything
here. Don't touch that.
Because I'd leave my DNA.
No, that's right.
The genetic database was conceived by 86-year-old entrepreneur James Lavoie Sorensen, a Mormon who doesn't give interviews and whose son James is in charge of the company.
Dad is a man of faith. He believes in God. He believes that we're all brothers, that we share connections.
Does he believe that our first parents are Adam and Eve?
He does believe that, yes. He believes that there is a genetic Adam and Eve that we're all descended from.
Researchers have collected DNA from 172 countries, over 90% of the world. Individuals gargle a mouthwash and sometimes take swabs. The company's global research has been recorded on video, from Austria to Australia and almost everywhere in between.
Just recently, we sent a team to Mongolia. Mongolia is a very important part of the world. We collected 3,000 samples with genealogies there. We're in the process of getting 600 samples from Iraq right now, 200 Shiite, 200 Sunni, and 200 Kurds.
They've already spent more than $40 million putting their DNA database together. So what prompted the company's founder to do this?
I think the hypothesis was that he could get any two people in a room and through this database show them how they were related and where they came from and how they belonged, and that this would change the way that they'd feel about each other, that instead of animosity, perhaps they'd feel a connection.
They've now launched a website, genetree.com, where individuals can order a DNA kit for $150. Once processed, they can begin discovering their own ancestry using a unique software program, a kind of DNA Facebook. I went through the process myself, producing saliva and swabs, and then packaging my DNA off to the lab in Salt Lake City. I'm about to learn what this database says about my own history.
You can watch kind of the growth of my lineage and how they moved across the world and how they came to the United States and really how I came to be here.
Jason Kessing, who was adopted at birth, is one of the first to do so.
I know my birth date, I know the place I was born, and nothing else. I was renamed as a baby and brought from California through the adoption service to Utah. That's all I know.
By matching his specific DNA to others within what's known as his haplogroup, that's those who share many of his genetic markers, Jason quickly made connections.
In one testing, I've learned already there are 5 exact matches for me. And coincidentally, one is in Ireland, one is in Bulgaria, one is in Australia, and 2 are in Utah.
In Utah?
I may have went to high school with direct cousins of mine, and they never knew it, and I never knew it. And eventually we find that I originated in Europe and then really right down in the Turkish region. We've always speculated. I have dark skin, so maybe I'm Italian. Maybe I'm Hispanic. You know, now I know who I am and why.
Are there any individuals, historic, famous, who might in some way be connected to my ancestry?
We all are so closely related, and so I would almost guarantee that you are related to famous people. You're famous yourself. So everyone who's related to you is related to a famous
That's true,
person.
actually.
We can put you on here.
Don't tell them. I was in for one huge surprise. In an effort to learn more about my own ancestry, I sent saliva and swabs to Sorensen Genomics in Salt Lake City. So what started as a sample of saliva in mouthwash ends up being processed, divided, defined, and accessible on the computer.
Yes.
As my DNA.
Yes.
And from that, you're able then to do matches in relation to other
individuals.
groups.
In one of these lanes, going across this way, would be one individual. There's probably about 90 individuals that are represented on that screenshot.
90.
Research scientist Natalie Reyes, who analysed my samples, walked me back through my own family history.
Those are my parents.
Those are your parents.
What does what you found out tell you about my history?
It's telling me that your ancient ancestors who originated somewhere between Northeast Africa and the Persian Gulf migrated southeast along the Asian coast and eventually sort of settled down in India. And that is then where your deeper roots originate. The second population are the Brahmins of Uttar Pradesh. Now the Brahmins, I'm sure as you know, are the highest caste in the Hindu social system.
I like what I'm hearing now.
The other group are the Rajputs of Rajasthan.
Yeah.
And they're also a major caste in India, which claim ancestry from ancient royal warrior dynasties.
Now we're getting somewhere.
So what this tells us is that you share ancestry with all these groups. We don't know yet if you descend directly from one of them.
Oh yeah, but come on. I may descend
yeah.
from the royal lineage of India.
But although this royal surprise was uplifting for myself, DNA can also cause an unwelcome shock.
So if a member of the Ku Klux Klan submits his DNA and suddenly discovers that in fact there was a black relative, you think that would be a good thing?
I think that could be a good thing, to realize that perhaps this hatred and this bias that has grown up through environmental influences there could be a realization that that would change that.
You'll know that the Nazi Party grew out of a public health movement. Do you think that gene tree could be seen in a similar light as something that's using science to discover the relative purity of people?
I think gene tree will do the opposite. It will enable people to see that they're related, that there isn't a so-called pure race, that we all have common roots.
Gene Tree has already had more than 36,000 people signing up to their site. There's no shortage of interest, but according to one expert, people should temper their expectations.
Professor Hank Greeley at Stanford University has said, and I quote, "A lot of genetic results are early, weak, and preliminary. One worries that people will think the information is more powerful than it actually is."
I think it's critical that people understand what it is that DNA can do and what it can't do. There are things that DNA cannot answer, and it doesn't happen nearly as quick as you see on television.
Well, I certainly feel I've been reconnected with my royal lineage. And I'm gonna start treating my producer like a slave from now on. How will I tell the difference? And that's our report for tonight. I'm Martin Bashir for Cynthia McFadden, Terry Moran, and all of us at ABC News. Good night, America.
Eden, do you have a question? Shoot.
Because everyone's DNA isn't sequenced, it's like that I understand that program, like it's more by association, but it almost seems like a hoax as well. Kind of like, oh, you're somewhat from this area, because I mean, everyone is originated from Africa and spread, and it's, I don't know, it just seems a little hoaxy.
So that's a really good question. And what we're going to do is talk a lot about that question on Thursday.